Right, where we left off, I've been talking about some discoveries in the 19th century, Schleiden and Schwann's discovery that all living things were made up of one or more cells, these living subunits that were fundamentally similar to each other in structure, even though they could be arranged to build a great many very complicated living things. And I talked about at the end, discovery by some dude named Schneider. His name's not that critical. He was looking at cells through the microscope, cells from a developing salamander tadpole, I think it was. And he was the first to observe something odd about the nuclei of these cells. Recall that normally when a cell is alive, but maybe not obviously doing anything, is just sitting there doing its metabolism and kicking back and doing whatever cells do, the nucleus just kind of looks like a round blob, maybe kind of grainy. In this drawing that he made over here on the left, the nucleus is this. There's a little dot in the center called the nucleolus, and we don't have to worry yet about what that does. Don't memorize that just yet. It's just there. But there's the nucleus. But when a cell begins to divide, the nucleus seems to turn into this set of little stringy things, like these things that I'm tracing right now. And the stringy things move in some kind of pattern, almost like country dancers. They line up in the center and then migrate to opposite sides of the cell. This is what they're doing here. And then group together again to form two new nuclei as the rest of the cell divides. Every cell comes from another cell, and every cell, to make more cells, goes through this division process. And when it happens, the nucleus turns into stringy bitskies, and these stringy bitskies do this funny little dance. Now, we can't go around calling these stringy bitskies because that just doesn't sound properly scientific, and we call them chromosomes. Chroma is Greek for colored, and soma is Greek for body. And they're called chromosomes not because they're colorful on their own, but because many of those stains we were talking about, these chemicals that, that color things, that stain them, stained the chromosomes especially strongly. In that cell, most of the cell is stained only a very light purple, but the chromosomes have very enthusiastically picked up the stain, and you can see they're a much more deep, vivid purple. So hence the name chromosomes, thingies that are colored. This picture is taken through a much more modern type of microscope called a scanning electron microscope. So this is not exactly what people saw in the 1870s because it hadn't been invented, but it gives you the idea that if you look at chromosomes at the very beginning of cell division, each chromosome is actually made of two parallel strands, and the strands are called chromatids. So these are two chromosomes photographed by the scanning electron microscope. And that red box there shows you one chromatid. And there's the other chromatid. Ooh, computer animation. I should be working at Pixar. There we go. So that chromosome is basically made of two strands, and they're stuck together more or less in the center. So there they are. And the larger chromosome next to it, I don't think I did the computer animation thing, but I'll draw it right now. There is one chromatid. And that is the other chromatid. Almost like taking two very long balloons and twisting them together, as you would if perhaps you were a balloon clown making balloons for a children's birthday party. So those are the chromatids right there, and the other chromosome is also made of chromatids. And the two chromatids are connected at a spot called the centromere. Mero is Greek for a division. So the centromere is just the central division. So that area right there, and that area right there that I've circled, is the centromere. So at the beginning of cell division, every chromosome consists of two chromatids that are stuck together at a point called the centromere that's often in the center of the chromosome, although there are plenty of cases where it's off center a bit. Okay. When a cell is not dividing, you can't actually see the chromosomes. As I mentioned, when a cell is just kicking back and texting on its cell phone, the chromosomes aren't visible, and the nucleus just looks like a blob, maybe a slightly grainy blob. But people noticed that if you watched a cell divide, you could count the number of chromosomes. And then when the cell was done dividing, the two cells would just kick back and the chromosomes would fade, and you'd get the grainy nucleus would reform. And yet, if you stuck around long enough and you watched those two cells divide into four, each of those two cells had the same number of chromosomes that the first cell had. These, by the way, are human chromosomes, with a few exceptions, one of which we will get to later. Every cell in your body's got 46 chromosomes. And in fact, the 46 chromosomes come in 23 pairs. We'll talk about that pairing later. Suffice it to say for right this minute that the fertilized egg that gave rise to you inside your mama's uterus had 46 chromosomes, and every cell that came from that fertilized egg, again, with a few exceptions like your red blood cells that we don't need to worry about, every cell that came from that fertilized egg, every cell that makes up you, as long as it's a normal cell and not a cancerous line, which I hope you haven't got, all of those cells have 46 chromosomes. And the same is true for other species. In horses, it's, I think, 64. In mice, it's 40. In fruit flies, it's 8. I don't remember a lot of other numbers, and it's really not important. It's just a number. But that number stays the same from one cell division to the next cell division. And that's evidence that the chromosomes are always there, even if you cannot see them when a cell is not dividing. It'll take us a little while to open this black box, but the chromosomes are always present. It's just that when division is not happening, they're not visible as, compact, individually countable units. And I will eventually explain where they are when you can't see them. So that dance that chromosomes go through when cells divide is known as mitosis. Mito happens to be Greek for a thread. So mitosis means threadosis. I hope you find that helpful, threadosis, because the chromosomes look a little bit like tiny pieces of thread. All right, mitosis is that thing that the threads do, especially when it's chilly, because when it's really chilly, mitosis is cold. That was a joke. I am imagining you all collapsing with laughter when I say mitosis is cold. Right. Okay. These pictures that you're going to see are pictures from it's not really important what they're from. These happen to be cells from a developing fish embryo. And that's a convenient place to observe this process. But the process in your own cells would look pretty much the same. There's some minor differences that we don't need to get into. Barring that, every eukaryote looks like this when it divides. So this is a fish cell that is not in the process of dividing. The nucleus is just kind of there. The chromosomes are present, but you can't really count them, and the nucleus just looks kind of like a grainy blob. And when a cell is not dividing, but just kicking back, we say it is in interphase. There are phases of mitosis, and the one before it starts is interphase, phase in between. Inter means between. When the cell begins dividing, for right now, we don't need to worry about what the switch is that triggers it to start dividing. We'll have to get to that after we plowed through some more material. But let's just say that the switch gets flipped. In the first stage of mitosis, the chromosomes start becoming visible. The nuclear membrane, which is what keeps the nucleus in and the rest of the cell out, disappears, and you start getting to where you can see the chromosomes. So the nuclear membrane was kind of right here where I'm drawing these dashes, but it's gone. And you can see there's little purple staining blobs in the middle of the cell. And this stage is called prophase, pro meaning first. So there's nuclear prophase. This, by the way, is a different cell, and it's been stained with a really cool technique called fluorescence microscopy. We can actually do this at UCA. We've got a fluorescence microscope. Some of the advanced biology majors get to learn how to use it. It's a pretty cool thing. What you're looking at here is a cell, but you can't see the entire cell. It hasn't been stained. What you're looking at is the chromosomes have been stained in this fluorescent dark blue. So this kind of blue cloud in the middle right there is the chromosomes. And green is showing you two tiny little organelles, two little structures called the centrosomes, start spinning a set of fibers made out of protein called the mitotic spindle. They are made of a type of protein called actin, which is also present in muscle. For right now, we don't need to worry about how they work. Let's just say that actin fibers are a kind of cable that's used in the cell when something has to move. And in this picture, most of the cell you can't see, but the chromosomes are in dark blue, and the mitotic spindle, or the spindle fibers, have been stained in green. So the centrosomes are right there and right there. And the spindle fibers are these things that I'm tracing out with the marker right there, attaching to the chromosomes, and they're going to do the work of moving the chromosomes around. These are hard to see with ordinary microscopy. You kind of can, but it's pretty subtle. But these pictures show what the spindle fibers look like much more clearly. At least I hope they do. Right. The spindle fibers do the work of moving the chromosomes around. You can think of them as being, the motor and the cables that move them. And the chromosomes line up in the center, and they line up along an imaginary plane. It's not a physical structure. It's basically an imaginary plane dividing that cell in two, called the metaphase plate. And so right there in the center, those dark blue things are the chromosomes lined up in the middle, and the spindle fibers are moving them into place. You can barely see them. This little dot there and there, those are those centrosomes, and kind of spreading out from them, you can barely see if you squint and clean your glasses and take lots of drugs, the spindle fibers kind of radiating out in all directions like that around the centromere. Again, it's not that easy to see under a light microscope, but the fluorescent picture shows them much more clearly. So there, again, the blue mess is the chromosomes lined up in the center, and the glowing green, you have the centrosomes there, and radiating out from them in all directions, you have the spindle fibers, making that green, looks like a pair of green stars, I suppose, green asterisks. That's the spindle fibers. Okay, now what? In the next phase, anaphase, the chromosomes break apart at the centromeres, and separate chromatids migrate to opposite sides of the cell. They're getting pulled by the spindle fibers. So there's one centrosome, there's the other centrosome, and the spindle fibers coming from each have made the chromosomes break at that centromere, and the separated chromatids are moving to opposite sides of the cell. That's anaphase. And there's another fluorescent micrograph. In blue, again, you have the chromosomes. They're stained blue. And the spindle fibers are what are dragging the chromosomes to opposite sides. Last phase, telophase. Telos is Greek for a goal. So this is the goal of the whole thing. The chromosomes clump into new nuclei, and they start fading. It's not easy to see exactly what's going on, but you start becoming unable to see them as individual little things under the light microscope. New nuclear membranes will form. So eventually you'll get a new nucleus there and there. And at the end, you've got two nuclei that go into interphase, and the cycle can repeat itself. And usually the cell itself divides at this time. In this whitefish cell, what happens is that the entire cell pinches in two. And again, I can't think of a better metaphor for it than if you were to take a balloon that wasn't inflated too much and kind of pinch it in the middle, you could end up with two separate bubbles. Eukaryotic cells will often just pinch off in two. And that's a process called cytokinesis. Kinesis means movement, as in, kinesiology is the study of human movement. You might know the word kinetic energy is energy that's involved in moving something. Kinesis is movement. Cyto is cell. Cytokinesis is movement of the entire cell. Technically, all those phases of mitosis that we were talking about refer only to the way the nucleus divides. Usually, cytokinesis happens at about the same time as telophase, and we end up with two completely separate cells. There's instances where this doesn't happen, and you end up with one cell that's got two nuclei. There are some organisms in which that's actually pretty normal. So they're not the same thing, and they don't always happen at the same time. But most of the time they do. Telophase goes with cytokinesis, which is the division of the entire cell. And that's what the spindle fibers look like at telophase. You've got two separated clumps of chromosomes, each one of which is now made of one chromatid. And, well, you can see the spindle fibers in green. I should mention that in animal cells and in many protists, when cytokinesis happens, the cell just kind of pinches in two. In plant cells can't pinch apart like that because they've got that stiff cell wall. Fungal cells, it's the same because they've got that cell wall. What happens is a little bit like what you would have to do in a building. If you're in, I don't know what room you're in, but let's say you're in a classroom and somebody decided that they wanted two classrooms, what they would do is build a wall down the middle. We actually had to do that in Lewis about 20 some years ago. We had to divide a large classroom into a couple of smaller ones. Sometimes we have to do that. And it's the same thing in plant cells. This is a plant cell in telophase. You can see the two clumps of chromosomes that are forming new nuclei. This fuzzy stuff right there that I'm outlining, that's the spindle fibers. And just faintly visible here is the beginnings of a new cell wall that will eventually extend all the way across and divide the cell. So in cytokine, okay, I seem to be back. Sorry about that interruption. Let's go right back to where we were. Okay. Okay, where we left off, when a plant cell goes through cytokinesis, a new cell wall forms and divides the cell in two. Okay, next slide. Here we go. Okay. So those are the stages of mitosis, and you're going to need to be able to recognize them. Interphase is the stage between mitosis when not much is obviously going on.
Mr. Wagner.
Yes.
We can't see the slides.
You can't. Okay, hang on. All right, let me fix that. All right. All right, let's go to, okay, I see there were things on the chat. Okay, so when I get knocked off, my screen share got knocked off. All right, sorry about that. Where are we? Oh, I'll tell you what, this is actually, I'm going to share something a little bit different because I can. It's possible to take mitosis and film it. And so this is a time-lapse video of what mitosis looks like. The actual process takes about an hour, but this has been sped up quite a bit. And I'll post a link to this if you want to see it again. So this would be prophase. The chromosomes are visible. Okay, here they are starting to line up in the middle. So we're getting towards, okay, lining up, lining up, lining up. Metaphase, anaphase has just started. There they go. And at the end of anaphase, we go into telophase. They're clumping together to form two new nuclei. Okay, here we go again. Here's some more. There goes anaphase, anaphase, anaphase.
Oh, okay. Well, it's been some
Okay, there's another video of mitosis. I think that's metaphase where they're lining up, lining up, lining up, lining up. There's metaphase, metaphase. Metaphase lining up, and anaphase starts in 3, 2, 1. There goes anaphase. There the chromosomes move to opposite sides of the cell, and there's telophase, and at the same time, cytokinesis. And now we've got 2 separate cells. I want to look at that first one just one more time. So in prophase, you can see all of the phases right there. Metaphase, they're moving, well, they're being moved by the spindle fibers, which you can't see in this particular picture, but they're lining up along that imaginary plane that goes from lower left to upper right. And boom, there goes anaphase, the separate chromatids moving apart. And right about there, we're in, whoops, I tried to get it to pause. And right about there, we are in telophase, where we've now got 2 clumps of chromosomes, and that line in the middle, I'm assuming this is a plant cell, that's going to be the new cell wall between the separate cells. So there's telophase and cytokinesis for you. That's what it looks like. And again, I will post the link to that if you want to see it again. Okay, right, back to the slides. Okay, at the start of mitosis, every chromosome consists of 2 chromatids. And remember, at anaphase, the centromere breaks. The separate chromatids move to opposite ends of the cell. So at the end, every chromosome consists of only 1 chromatid. So we go from chromosomes that are made of 2 chromatids to chromosomes that are made of 1 chromatid. So at the start of mitosis, each chromosome is going to have a centromere that connects up 2 chromatids. And at the end of mitosis, every chromosome consists only of 1 chromatid. But if you were to watch that cell divide, somebody mute, please. If you were to watch that cell divide and watch the 2 daughter cells and keep them alive under your microscope slide and watch them until they divided again, each daughter cell is going to start with 2 chromatids per chromosome. So in mitosis, we go from 2 per chromosome to 1 per chromosome, but somewhere in interphase that we cannot see, each chromosome sprouts a new chromatid somewhere in interphase. We can't see yet how they did it. We will get to how they did it soon enough, but right now that's a black box. So the way we now think of cells' lives is what we call the cell cycle. Mitosis is, of course, divided into, there's prophase, there's metaphase, there's anaphase, there's telophase, and at the end of telophase, the 2 cells go into interphase. We now divide interphase into a stage called G1, or first growth phase, where the cell just grows and does whatever it's doing and runs its metabolism and all that. An S phase, which is what we can't yet see, where the cell's chromosomes sprout new chromatids. The chromosomes are still there, they're just not visible under the light microscope. And then G2 comes after S. The cell grows some more and prepares for going into mitosis again. So we divide interphase into G1, S, and G2. If anybody's curious as to how long these periods take, it actually depends on the cell you're talking about. Mitosis roughly would take about an hour. G1 could take anywhere from a few minutes to many years, depending on what cell you're talking about. There are some cells where G2 takes almost no time and you go from S almost immediately into mitosis. There's others where G2 might just pause and last indefinitely. It all depends on the type of cell. A rough average would be that mitosis takes about an hour and interphase might take maybe a day, but that varies immensely, depending on what organism you're talking about. So we divide interphase into G1, S, and G2, and we cannot yet clearly see exactly what's going on in G1, S, and G2. S is the mystery phase where the chromosomes sprout new chromatids. Who cares? Single-celled eukaryotes reproduce by mitosis. That's how they make more of themselves. Development is driven by mitosis. You went from a single fertilized egg cell in your mama's uterus to the roughly 37 trillion cells that make you up now because of a heck of a lot of mitosis. You went from being one cell to 2 cells to 4 cells to 8 cells to 16 cells, ultimately to 37 trillion. Hang on. Okay, just somebody typing a name. Okay. And you keep carrying out mitosis today because every day you lose 50 to 70 billion cells. They die. The cells that line your cornea, that's the part of your eye that you put the contact lens on, are constantly dying and washing away in your tear fluid. The cells that line your intestine are constantly dying and being shed in your feces. The cells in the outer layer of your skin are constantly dying and flaking off and forming dandruff. And to keep you from wasting away completely, they all have to be replaced by new cells. So in many parts of your body, mitosis is still very much active, replenishing cells that have worn out and are being broken down and discarded. Mitosis has to happen whenever you get a wound. The scar that forms on a wound is made of specialized cells called fibroblasts that move into the wound and divide to regrow scar tissue that holds you together. And then finally, I'm not prepared to talk in detail about this just yet. Maybe we can a little bit later. But let's just say that cancer is basically mitosis with the brakes off and the accelerator stuck. A cancerous tumor is a group of cells that are actively dividing, they're undergoing mitosis, and they can't stop and they can't be controlled. That's what a tumor fundamentally is, a bunch of cells that are undergoing mitosis rapidly and like Lionel Richie, they can't slow down. Don't know if any of you remember Lionel Richie, post-millennials, right? So that's what you need to know for right now about mitosis, and we'd observed it starting in the 1870s. It gets worse because there's actually a second type of cell division that eukaryotic cells undergo. Dun, 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 dun. It's called meiosis. Now, what's the difference? In you and in animals, meiosis is used in one context only. Mitosis is how you develop. Mitosis is how you go from one cell to trillions. It's how you repair. worn out body parts and fix wounds and all of that. In animals, meiosis is used for one and exactly one thing. It's how you make egg cells or sperm cells, or in many organisms, both at the same time. Like if you're an earthworm, you would use meiosis to make both at once. Now, to understand meiosis, I've got to give you some background on chromosomes. When you look at chromosomes really carefully, the chromosomes are not all identical. Some chromosomes are long, some chromosomes are short, some chromosomes have the centromere close to the actual center. So chromosomes, there's two chromatids. And chromosomes where the centromere is close to the actual center are called centric. So there's my drawing of a centric chromosome. But you can also find chromosomes where the centromere is off center, and we call these acentric chromosomes. And chromosomes are also different in the pattern of bands that you can see on them with staining techniques. Exactly what these looks like depend on how you've stained them, and there's several ways to do this. But when you stain chromosomes with the right stain, you'll find that they've got what look like little bands or stripes on them, and they vary a lot in exactly where those bands are. Different chromosomes have different patterns of bands. And it turns out that if you take a bunch of chromosomes from one cell and you try to classify them, you maybe take pictures of these chromosomes and start moving the pictures around. It turns out that in a typical eukaryotic cell, there are 2 of every chromosome type. And a cell that's got 2 of each type of chromosome is said to be diploid. Diplo is Greek for double. So diploid means that they are double-oid. They've got 2 of each chromosome type. And here's an example. These are chromosomes from a human female. They have been stained with a fluorescent stain in which different areas of each chromosome stain either red, orange, or yellow. And the chromosomes here have been moved into pairs, and you can match them up by length, by centromere position, and by banding pattern. You know, kind of like if you're playing Old Maid, if anybody ever played that. And in normal humans, we have 46 chromosomes, and when we sort them, we get 23 pairs of chromosomes, which we just number 1 through 22, and there's a pair right here that's not numbered. It's a pair of X chromosomes, and we'll talk later. We will get to on Thursday what that X means. That's got to do with sex determination, but we will get to that soon. Don't worry about it for right now. What you need to see is that in a normal human, you've got 23 pairs. And this is how you can tell that they're paired. You can match them up by centromere position, banding pattern, and size. Okay, there we go. Different animal and plant species have different chromosome numbers. These are mouse chromosomes. Typical mice will have 20 chromosome pairs per cell. That's 40 chromosomes in all. And again, we can match these up by the banding pattern. This one was not done fluorescently, so I'm sorry it's kind of drab. But you can see members of the same pair have the same set of bands. Now, aside from the fact that usually closely related organisms have similar chromosome numbers. Domestic horses, I think, have 64, and donkeys have 66. You've got 46, and chimps have 48, and so on. Other than that, there is no simple relationship between how complex or how, in quotes, quote, quote, advanced an organism is and how many chromosomes per cell it's got. It's just a number. The organism with the largest number of chromosomes has 1,260 of them arranged in 630 pairs. And it is this dinky little thing called the snake tongue fern that you could step over and never even realize was there. It's not an organism that really draws attention to itself in any way. You know, it's not a huge tree or anything like that. There's plenty of animals that have more chromosomes than we do and animals that have fewer. It's just a number. Not really important why the number is what it is, not for our purposes. But animals, plants, and many other eukaryotes form specialized cells for sexual reproduction, and those are called gametes. In many protists and fungi, all the gametes are the same size, and you can't tell them apart by looking. We call them, in some cases, we might call them plus and minus gametes, or A and alpha gametes, or various other things. But in animals and plants, gametes come in two forms, big and small. And the big gametes are called eggs, and the small gametes are called sperm. And the only important difference for our purposes is that eggs are bigger. Sperm are smaller. And the thing about gametes is that they've only got one of each pair of chromosomes, one of each pair. And we call this being haploid. So gametes are haploid, not diploid. I should mention, by the way, that in plants, fungi, and some protists, this can get a little bit odd, and we probably won't have time to get into why. But in animals, certainly in you and in all other animals, the only cells in your body that are haploid are either your eggs or your sperm, depending on your biological sex. Or if you're an earthworm or a Caribbean butter hamlet fish, excuse me, you might have eggs and sperm in your body at the same time. That's called simultaneous hermaphroditism, and it's not all that uncommon. Oh, the really cool thing, there's a number of organisms that will start out producing one type of gamete and then switch. They will switch biological sexes. Clownfish, for example, start out as male and then switch to female later in life. So if Finding Nemo was at all realistic, Marlin would be trying to raise his son Nemo while at the same time figuring out how to handle the fact that he's in the process of transitioning to Marlena. And I wish Pixar had gone with this. It would be a much better movie, you know, and I think much more, you know, socially relevant if they'd worked the fact in that Marlin is supposed to turn female, because that's what real clownfish do. That's not at all uncommon. There's a lot of fish that will start out as one sex and switch to the other. And I've just ruined one of your favorite movies. You're welcome. Dory would be okay, by the way. They don't do that. Blue tangs don't do that sex change thing. Right. So gametes are typically either eggs or sperm, except in many protists and fungi. And gametes are haploid. They've only got one of each pair of chromosomes. And the process of cell division that produces haploid cells from diploid cells is called, meiosis. In animals, this is how we make eggs and sperm. Meiosis is going on either in the seminiferous tubules of your testes or the follicles of your ovaries, depending on whether you happen to have ovaries or testes. So in animals, this is how we make eggs and sperm. Plants, things get a little bit weird, and we probably will not have time to go into alternation of generation in plants. So I'm trying to keep this as smooth as I can. The basics of this, by the way, were worked out by this Belgian guy in 1883. And another name for meiosis, by the way, is reduction division, because with mitosis, you begin with a fixed number of chromosomes, and if everything goes right, you end with the same number of chromosomes. One of your cells undergoing mitosis will start with 46 and end with 2 cells, each of which has 46. In meiosis, by the time it's over, you reduce the number of chromosomes per cell by half. Every cell in your body's got 46 chromosomes, except your eggs or your sperm, which have 23. Division exactly in half. So that's a human egg cell photographed with the scanning electron microscope with a whole bunch of human sperm cells that are trying to burrow in. One sperm cell will make it, and as soon as one sperm cell gets in, the egg undergoes a change to the egg cell membrane that blocks all other sperm from getting in. So only one is going to be able to fertilize that egg. Now, the egg cell has a nucleus inside. You can't see it, but it's there. And the egg cell nucleus contains 23 chromosomes. Every sperm cell has a nucleus. In fact, a sperm cell basically is a nucleus with a propeller, this little tail that it beats to swim. There's not much else in a sperm cell. And every sperm cell nucleus contains 23 chromosomes. So that's an egg cell just after fertilization. That's probably the egg nucleus right there. There's the sperm nucleus right there. They are joining and fusing and becoming one. Da, 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 da. And the fertilized egg is called a zygote. If you're curious, remember, again, I'll never test you on the Greek and Latin roots, but it might help you learn the vocabulary. Zygo is Greek for a yoke, and a yoke is a piece of wood that links together a pair of horses or a pair of oxen that are pulling a carriage or a plow. Okay, a yoke is something that links two things together. And a zygote is thus a thing in which two things are yoked together. That refers to the joining of the nucleus from the sperm cell and the nucleus from the egg cell. And the fused nucleus now has 2 of each chromosome, in this case 46. So the unfertilized egg had 23, the sperm cell had 23, the zygote has 46, and the zygote will divide by mitosis and produce every other cell in your body. Almost all of those will have 46. By the way, if there's an error in meiosis, you can end up with an egg or a sperm that doesn't have 1 of every chromosome. It can either have 2 of one of the chromosomes, or it can be missing 1. These are the chromosomes from someone who's got 3 copies of chromosome 21. Whoops. Okay, I'll circle that to make it permanent. Chromosome number 21 exists in 3 copies. This person's, okay, the egg cell that gave rise to this person had 1 of each, and the sperm cell had 1 of each, except either the egg or the sperm, it doesn't really matter which, happen to be packing 2 copies of chromosome 21. So this person ended up inheriting 3 copies of chromosome 21, 2 from one parent and 1 from the other. And the reason I'm bringing this up is that people with 3 copies of chromosome 21 have a condition called Down syndrome. Developmental delay, distinctive physical appearance, higher risk of heart defects, and various other things. Down syndrome is fairly common. I'm sure you've seen people that have it, and it is a result of an error in meiosis when either the sperm or the egg that gave rise to this person was being formed. So one reason why it's important to understand what goes on in meiosis is that if it goes wrong, you can have a child born with a missing chromosome or an extra chromosome. But if nothing goes wrong, then you inherit 1 of each chromosome from mom and 1 of each chromosome from dad. Okay. I'm not going to force you to learn every last step of meiosis. I'm going to try to break this down as simply as I can. Meiosis is actually a double division, and we divide it into meiosis 1 and meiosis 2. The end result is 4 cells at the end. So if you are a man, your cells inside the little tubules in your testes called spermatogonia are constantly dividing by meiosis, and each spermatogonium cell produces 4 sperm cells. If you are biologically female, it's a little bit different, because when you're making egg cells, cell division is actually not even. And each oogonium cell, when it divides by meiosis, ends up producing 1 egg cell and 3 little castaway things called polar bodies. So there's a little bit of a difference between the way that biological males and females do it. For our purposes, we're not going to worry about that, but you might encounter this if you go on to further study. So before meiosis, a cell goes through the G and the S phases that we talked about. So when meiosis begins, it's got a pair of every chromosome, and every chromosome consists of 2 chromatids. So right there is a pair of chromosomes, and each chromosome is made of 2 chromatids. In meiosis 1, each daughter cell gets 1 of each pair of chromosomes. So each daughter cell has 1 chromosome that's made of 2 chromatids. And then at the end of meiosis 2, each daughter cell has 1 chromosome, 1 of each pair of chromosomes, and each chromosome consists of 1 chromatid. So we go from 1 cell that's got 2 of each chromosome, each of which has 2 of chromatids, to 2 cells with 1 of each chromosome, and each chromosome has 2 chromatids, to 4 cells with 1 of each chromosome, and each chromosome has 1 chromatid. So there you have it. Before meiosis, we start with 1 cell, either a spermatogonium in your testes or an oogonium in your ovary, depending on your biological sex. Each of those cells contains 2 of each chromosome, and each chromosome is made of 2 chromatids. That oogonium or spermatogonium divides one time, and at the end of meiosis 1, you've got two cells. Each cell has one of each chromosome, and each chromosome has two chromatids per chromosome, with the wrinkle that if you're making egg cells, the division isn't even. So instead of dividing into two equal cells, you divide into one egg and one tiny little thing called a polar body. And then they divide again. And at the end of meiosis 2, you've got 4 cells. Each one of those cells contains one of each chromosome, and each contains one chromatid per chromosome. Hence, it's reduction division. It's a double-stage division. And if it works right, you divide the number of chromosomes in half at the end of meiosis. I've got some pictures. I'm not going to talk you through every last stage. These pictures, by the way, are actually from lily anthers. They show nuclei in developing pollen grains. But the process is not grossly different in your own gonads or really anywhere in eukaryotes. Prokaryotes, by the way, don't do any of this. We will be able to talk later as to what they actually do. But bacteria and archaea don't have chromosomes, at least not that have this shape. We will talk later about what they have got. So anyway, this is flower pollen developing, but your own cells don't do anything especially different. Meiosis 1 starts out looking like mitosis. So on the left, you've got early prophase 1 with the chromosomes just barely visible. And on the right, you've got late prophase 1 where the chromosomes are now very clear and nice and thick. But there's something else that happens at the beginning of meiosis 1. Each chromosome pairs up with the other member of its pair. So this and that, those are pairs of chromosomes. And the paired up chromosomes undergo crossing over, which means that they break and exchange equivalent pieces. In lecture, I would demonstrate this in person. I'd get somebody about my size to stand next to me. And we can't actually do this. But if I were to stand next to somebody who is my size, and we were to amputate our arms at the elbow and then switch so that I had his arm and he had mine growing on our bodies, that would be what crossing over is like. In crossing over, paired up chromosomes exchange pieces. And under a very powerful light microscope, it looks like this. This is a pair of chromosomes, and these little X-shaped points right there, and there, and there, and there are where chromatids are breaking and getting swapped. Anyway, that happens, and the paired up chromosomes move to the midline and they pull apart. And meiosis 1 ends with the formation of 2 haploid cells because one of each chromosome moves to each daughter cell. This would be anaphase 1. And at the end of it, you have 2 cells. Each one has one of each chromosome, but each chromosome consists of 2 chromatids. And then we go into meiosis 2, the second division of meiosis. And this works exactly like mitosis does. The chromosomes line up and they separate at the centromeres, and each chromatid moves to opposite ends of the cell. So there we have 2 cells that are caught in the act of meiosis 2. This looks like it's anaphase 2. And the end result is we get 4 haploid cells. So there we've got 4 haploid cells, each of which has one chromatid per chromosome. And this is like telophase 2. That's the end result that we get. And again, that was lily pollen, but what your own gonads are doing doesn't really look that different. This is pretty much the same in all eukaryotes. Okay. I'm about to finish. This is the last slide, but this is important. This part often seems like drinking from a fire hose. I've just thrown a lot of detail about Mendel crossing a bunch of stupid pea plants and a bunch of cats with funny looking fur, one of which I'm looking at right now. And I've just thrown you a bunch of that. And now I've just thrown you a bunch of detail about a bunch of stupid little thread-like things and a bunch of stupid cells that do this dance, and they come together right now over me in the middle of the cell, and then they pull apart and the chromatids separate and they do this and they did that. Why am I going through all this crap? Okay. Mendel had worked out that genes come in pairs, right? You inherit one of each pair of genes from each parent, and you will pass on one of each pair to each of your offspring, right? That's basic to Mendelian genetics. Meiosis works the way it does because cells produced by meiosis have one chromosome of each pair. You inherited one of your copies of chromosome 1 from your mom, and the other copy came from your dad. You inherited one of your copies of chromosome number 2 from mom and one from dad. You inherited one copy of chromosome number 3 from your mom and one from your dad, and so on. You got one of each pair of chromosomes from each one of your parents, and you will pass on one of each chromosome to each of your offspring if you have them. And this must be critically important because, as we mentioned, if meiosis goes wrong, an egg or a sperm can end up with a missing or an extra chromosome. Very often, this means that a zygote that has an extra chromosome or a missing chromosome, usually it doesn't develop at all. Usually it ends up miscarried. In humans, this often happens before the woman even knows that she's pregnant. An estimate of roughly half of all conceptions miscarry. Usually you never know. It's just one of those things. And a leading reason why they miscarry is that they have extra chromosomes or that they're missing some. Only in a few cases do zygotes with extra or missing chromosomes develop at all into babies. And those babies usually have medical conditions like Down syndrome. And there's a few others that they can have. So this has to be really important, making sure that you get only one of each chromosome from each parent is critical. That's why meiosis looks the way that it does. You're shuffling your chromosomes in a very precise way to ensure that every egg or sperm is carrying only one of each pair. That's why that matters. And think about this. Why are genes and chromosomes following the same rule? It might have seemed like I'm throwing a lot of jigsaw puzzle pieces at you folks, but this is where the pieces ought to start coming together. Mendel worked out, just by doing arithmetic on his p's, that genes come in pairs and one of each pair is passed to each offspring. Microscopists in the 1800s worked out that chromosomes come in pairs and one of each pair is passed to each offspring. Aha! Why are genes and chromosomes following the same rules? Okay, you ought to have the mental sensation of jigsaw puzzle pieces beginning to come together with a satisfying click. But to answer that question, we will have to wait till Thursday. So I will now stop the recording.